Hello everyone, and welcome to episode 11, Remastered. In this series about the cell, I've been talking about cellular structure, organelles, how cells produce energy, all this cool stuff. And so today, I'm going to be discussing how cells interact with each other, how they communicate and group together to form communities like tissues, and subsequently how these form entire organisms. In order for any organism to survive, its cells have to be connected to one another both physically and chemically. Physical connections allow groups of cells to maintain a continuous form without randomly falling apart like a pile of rocks. To pathogens, your skin is like a sealed barrier because of these cellular connections. Now, chemical connections are not mechanical, but they're just as vital to the survival of the group of cells. Chemical signaling and regulation is required for groups of cells to manage all of their shared resources. Inefficient processing and distribution of nutrients and oxygen, inefficient waste management, or chronic hormonal misfiring, all of these will harm and eventually kill the tissue or the entire organism. For example, when you belt a tree, you effectively kill it. By removing the outer ring of tissue, you starve the rest of the organism directly above that ring. The roots will collect water and mineral nutrients, but belting the tree removes an outer strip of that vascular tissue, and it basically destroys the nutrient input route for all of the cells further up the tree. It breaks that pressurized mechanism that the tree uses to move water up its veins. Its cells will then starve and die, and the whole tree will follow shortly afterwards. Consider that the cellular membrane is the barrier that separates life from non-life, the organized interior of the cell versus the chaotic and dangerous exterior. The cellular membrane is thus the interface between life and non-life, and it's upon this interface that the physical connections between cells are built. This membrane interface is heavily studded with proteins, with pores, active transporters, glycoproteins for signaling, all sorts of stuff. Most cells also secrete a series of compounds into the exterior of their membranes, which forms a flexible shell or a coating. It's protective. These compounds are called fiber composites, because much like the structural carbohydrates that I covered in episode 5, these fiber composites are composed of fibers, which can pile together and link together or twist together to form sheets and other large structures. These fibrous sheets are covered in a surrounding material called a ground substance. This is very easy to understand in terms of concrete. Concrete is laid down as a very heavy, kind of liquidy goop, which then dries and solidifies. Steel rods are often crisscrossed within the volume of a concrete block, and the wet liquid concrete is poured around these steel rods. The concrete resists compression, like heavy objects can put a lot of pressure on the concrete and it'll be just fine. But concrete isn't as good at resisting tension. The steel rods are put in because they can resist the tension of being pushed or pulled. And together, these qualities make the concrete block a very sturdy and useful item. The fiber composites in the cells are very similar. The fibers give the cell a degree of resistance from tension forces. They give it a little bit of flexibility, while the ground substance provides resistance to the pressure of all of the adjacent cells. The particular types of fibers and ground substances that are used by cells have various strengths and weaknesses and qualities that make them better or worse at specific functions. Animal cells, for example, secrete flexible compounds that can stretch or bend, and this makes sense because animal cells often expand or shrink. Plant cells, in contrast, secrete compounds that can form stiff, rigid tissues, like wood that can support tremendous pressure. In plant cells, this extracellular layer of fiber composites is a carbohydrate-rich structure called a cell wall. Young plant cells produce a primary cell wall, which is composed of crisscrossing bundles of cellulose fibers called microfibrils, and together these are submerged into a gooey ground substance of sugars called pectins. The microfibrils and the pectin goop form a very strong sheet all around the plant cell. The pressure of the water within the cell, called the turgor pressure, causes the membrane to get pushed up against the inside of this cell wall. In this way, the wall defines the shape of the plant cell. Some plant cells may, as they grow older, produce a secondary cell wall, which is secreted in a layer between the membrane and the primary cell wall. The compounds in the secondary cell wall will vary enormously, not just between species, but between tissues within a single organism. The secondary cell wall in wood tissue, for example, is lined with an extremely strong and durable class of polymers called lignins. The secondary cell wall in leaf tissue is lined with a wax that greatly assists in reducing water loss. The secondary cell wall in stem tissue is stiffened with additional layers of cellulose, which provides greater structural support. If stem tissue was as sturdy as leaf tissue, for example, most plants wouldn't be able to hold themselves upright or move water from their roots to their leaves or their flowers. The cells in their stems have to have this strong structural support to maintain the plant's physiology. Now, animal cells don't possess a cell wall, and that's good, because if we did, we probably wouldn't have the same level of mobility that we have now. We would have a level of mobility on par with plants. Now, instead of a cell wall, animal cells will secrete a much more flexible substance called the extracellular matrix. Where the walls of a plant cell are carbohydrate-rich, the extracellular matrix of animal cells is protein-rich. The polymers in animal cells are protein fibers called collagen. The gooey ground substance that encases the collagen is composed of a swamp of proteoglycans, which are like nuggets of proteins coated with numerous larger carbohydrates, or sugar molecules. As in plants, the cells within animals also show a variety in how their extracellular matrix is expressed. In skin cells, there's relatively little extracellular matrix separating the cells. Instead, they're connected directly through tight junctions and desmosomes, although I'll discuss those more in a few minutes. In tissues like bone and cartilage, there's a lot of extracellular matrix separating the cells. Now, animal cells are connected to each other through this extracellular matrix. Cytoskeletal components on the inside of the cell form linkages to membrane proteins called integrins, which attach to a class of rope-like proteins called laminins. These laminins interlink with one another to form a web or a net-like lattice that covers the exterior surface of the cell. This laminin net perforates and climbs through a layer of proteoglycans, ultimately connecting to the larger collagen fibrils that run alongside multiple cells. Animal cells are typically composed of millions of these structural collagen fibrils, collectively providing strength and great flexibility. So you have these animal tissues, these masses of animal cells that are all unbounded by a cell wall, so they're all soft and flexible. And so to keep them all together, to keep them organized, you have this mesh-like net of protein fibers that holds them all together. There's other mechanisms for cellular connection besides simply getting tied up to the same big net. These other connections are particularly important in epithelial cells, which are flat, thin cells that usually exist on the surfaces of organs or the lining of blood vessels, to form these smooth, sealed layers. Like the bilayer membrane is to the organelles within a cell, epithelial cells are to the organs within an organism. These cells form barriers within which specialized environments can be created. And this is really important because you wouldn't want your stomach acid leaking into your liver, or air from your lungs leaking into your heart. Both of these problems would cause you serious pain, and would most likely kill you. As you can see, it's very important for epithelial cells to have tight connections so that they can form these sealed layers. Two adjacent cells will typically have some sort of chemical adhesive holding them together. In plants, the space between the walls of two adjacent cells is called the middle lamella. Now within this middle lamella, there's a gel-like layer of pectins that holds the two cell walls together, much like bricks and mortar. In animals, this space is filled with a layer of goo, rich in proteoglycans and polysaccharides, which is also adhesive, but a little more flexible. Now, because animal cells don't have thick walls separating them, the animal cells can be grouped together much tighter, much closer to one another. This also allows for the formation of direct structural connections between adjacent cells. Remember those tight junctions and desmosomes that I mentioned earlier? Well, those, as well as gap junctions, are some of the direct structural connections between animal cells. So let's start with tight junctions. A tight junction is a region where the cells are connected very close together. They're pressing up against each other. Specific membrane-bound proteins on each cell will connect with one another to form wavy rows of interlocking protein pairs. These rows of proteins are kind of like stitches between two pieces of fabric, or like a row of staples connecting two pieces of paper. These tight junctions are, indeed, quite tight, often forming a waterproof seal between cells. But the tight junctions are weak, and they can be easily broken. Now, this might sound like a bad thing, but it's actually really important for cellular function, because it means that tight junctions can be regulated. Loosening the tight junction encourages the movement of particles between the cells, while tightening the tight junction encourages the movement of particles across the cells. Your blood vessels and bladder, for example, generally keep their tight junctions very tight. Otherwise, they'd leak, and you would have internal bleeding, or you'd have your urine just spilling all over your pelvis. Your intestinal cells, on the other hand, will regularly loosen their tight junctions. They do this during digestion to aid in the absorption of nutrients, and then they retighten afterwards. By loosening up these tight junctions, the nutrients can move past the epithelial layers on the inside surface of your intestines to get absorbed by cells that are deeper in the tissue, and this improves the total efficiency and effectiveness of digestion by greatly increasing the surface area of your intestines. Now, desmosomes offer a significantly stronger connection between cells than that provided by tight junctions. Where tight junctions are similar to rows of staples holding two pieces of paper together, desmosomes are like groups of iron bolts holding steel plates together. Cells of a similar tissue type will connect together through their desmosomes, which are composed of three distinct parts. On the inside of each cell membrane sits an anchoring protein, which is kind of like a plate or a washer. Each anchoring protein is then bound to a large number of smaller membrane proteins. This cluster of membrane proteins reaches through the membrane up into the space outside of the cell, where they then connect with the other membrane proteins on the anchoring plate of an adjacent cell. These membrane proteins are called cadherins. They're responsible for the specificity of bonding between cells and for the relatively high strength of the desmosome connection. Okay, so we have the anchoring proteins and we have the cadherins, which leaves the third part, which are the intermediate filaments. On the inside of the cell, loosely packed masses of intermediate filaments will connect to the anchoring plate, and they'll run through the cell, through the interior of the cell to the other side, to reach another anchoring plate that's somewhere else on the membrane. In this way, the intermediate filaments connect the desmosomes in a network that is simultaneously inter- and intracellular. When a force tugs on one desmosome, the intermediate filaments will spread out that force to all of the other desmosomes connecting to other cells. This provides a consistency to the organization of the cells within the tissue, as well as a physical consistency to the tissue itself. Because the desmosomes are really tightly anchored into the membrane, if there's too much force pulling on them, they can just be ripped right out of the membrane, and that'll create a giant hole in the cell, and that can most likely kill it. So to prevent this problem, all of the bolts are held together on the inside of the cell through all of this wiring, through all of these intermediate filaments. And so if something pulls really hard on a desmosome on one side of the cell, it'll start pulling on all of the other desmosomes all across the cell. And so this will prevent the cell's membrane from being ripped apart. The last type of direct cell-to-cell connection that I'm going to discuss is not so much structural as it is communicative in purpose, and these are gap junctions. The gap junction is a group of pores in each cell's membrane, and these group of pores can align with one another to form tubes connecting the inside of one cell to another. These tubes penetrate the membranes of both cells, and so this allows them to exchange small molecules and ions. In this way, adjacent cells within a tissue can communicate rapidly with one another by sending hormones or nucleotides through the tubes. When the cells can communicate quickly in this fashion, they can coordinate their behavior. Consider your cardiac muscle cells, the cells in your heart that allow it to pump. If each cell contracted on its own, your heart wouldn't contract to an irregular beat. It wouldn't contract at all. The individual contractions of the individual cells would make your heart look like a quivering, squirming mass, unable to forcefully pump blood. Now, without going into too much detail, your heart has a specialized group of cells called the Sinoatrial Node. This node produces an action potential, which is sent to every cardiac cell, depolarizing their membranes and causing an influx of sodium ions. The gap junctions allow the sodium ions to be rapidly distributed across the cells of the heart, coordinating their contraction into a regular pulse. This coordinated contraction enabled by the gap junctions allows your heart to create regular, forceful pumps, which then circulates your blood and keeps you alive. Okay, so there's gap junctions, there's tight junctions, and there's desmosomes, and these are the close physical links between adjacent cells. And this leads one to wonder, how do cells interact when they are far apart from one another, when there's no physical connections between them? Multicellular organisms like humans and pine trees and elephants, and even smaller organisms like mosquitoes, parasitic worms, and even multicellular microbes like tardigrades and rotifers are all huge when you look at them from the perspective of an individual cell. Cells are really, really tiny, so from their perspective in a multicellular organism, the distance from one end of the body to the other is tremendous. So how do these distant cells from one end of the organism to another, how do they communicate with each other? How do they interact as part of a single, unified organism? The answer? Signaling molecules. The concept of signaling molecules is pretty basic. When a cell or a tissue needs to send a message to another cell or tissue, the sender will secrete a particular molecule. This molecule travels through the body, typically using the vasculature in plants and animals, although some smaller organisms that don't really have vasculature will just use diffusion to move the signal molecules through their tissues. This signal molecule will reach its target tissue, where it will bind to some kind of specific receptor and cause a reaction. This molecule is a signal molecule because it's sent from one place to another place, where it reacts with cellular machinery to initiate a particular response. The message is received when the target tissue reacts to the signal molecule, stimulating a particular behavior, or depolarizing the membrane, or initiating a chemical cascade, or doing whatever it is that the activated cell or tissue does. If you happen to be a fan of George R.R. Martin and his book series, A Song of Ice and Fire, you might appreciate this analogy. The signaling molecule will go from one cell to another, and it'll cause a particular response that depends on both the specificity of the tissue that's receiving it and the specific molecule that was sent out in the first place. Now, in George Martin's book series, there's a place called the Citadel, which is home to a group of scholarly monks. The seasons of Martin's world are irregular and unpredictable, with winters and summers lasting sometimes for years, maybe even a decade. When the summer ends and winter begins, those monks at the Citadel will send out a white raven to the castles of Westeros, and the white raven acts kind of like a signaling molecule. Once the castles receive this white raven, if they haven't noticed the weather yet, the nobility will typically begin to make preparations for the potentially years-long winter. So the Citadel is the source of the signaling molecule. The white raven is the signaling molecule. The castle is the receptor cell, or the receptor tissue, and the rookery in the castle, where the raven comes to land, is kind of like the cell receptor, where that particular signaling molecule gets bound and detected. When the bird keeper in the rookery takes the message to the castle nobility, the signaling molecule will bind to its receptor and initiate a response. The nobility will begin preparations for winter. The cell response has begun, and the signaling is complete. Perhaps the most well-studied and well-understood group of signaling molecules are the hormones. Hormones are small chemicals that are secreted into the bloodstream, where they then reach and interact with one or more target tissues to cause specific responses. Hormones are very powerful molecules. They need only be present in a very small concentration to cause a very potent response. There's two large groups of hormones. They're either hydrophilic or hydrophobic. The hydrophobic hormones can interact with and pass through the equally hydrophobic phospholipid bilayer of the cellular membrane, and this allows them to bind to receptors inside of the cell or an organelle, or even receptors inside of the cell's nucleus itself. Hydrophilic hormones can't pass through cellular membranes, and so they only bind to receptors on or outside the surface of the cell. The receptor is fundamentally important to cell signaling, as important, or arguably more so, than the hormone or the signaling molecule itself. When a hormone is released into the bloodstream, it'll go throughout the entire body. It comes into contact with everything. It goes everywhere that the vasculature takes it to. However, the only tissues that actually bind and react with the hormone are those that have the right receptor for it. When multiple tissues have cells that express a receptor for the same hormone, then each tissue will bind to the hormone whenever it's released. In this way, a single hormonal release can activate or stimulate responses in multiple tissues simultaneously. The cell's expression of receptors can change over time. Cells that are exposed to very low levels of a particular hormone may express more receptors in an attempt to capture any hormone molecules that it can. Conversely, cells that are exposed to a high level of a particular hormone will begin to express fewer receptors, because as the cell is just swamped in the hormone, there's a certain reliability that the cell signal response will still be thoroughly stimulated even if there's fewer receptors for the hormone to bind to in the first place. In cases like this, where a receptor experiences a high frequency of stimulation, it might find its ability to respond to the hormone gradually degrade over time. This happens in the brains of people who are addicted to drugs, like caffeine, cannabis, alcohol, nicotine, opiates, and virtually everything else. Prolonged use of these substances creates a tolerance, which means that the person has to take more of the drug to achieve a similar effect. This is why some people need 3 cups of coffee to function properly, or 5 or 6 beers before they even start feeling a buzz, or 2 joints before they start to feel alright. Receptors can also be blocked in a form of competitive inhibition. Some medicines and drugs work by competitively binding with these receptors, while others work by blocking the receptors entirely, which prevents them from being activated in the first place. So after a signaling molecule has been bound to a receptor, what does it mean to say that a response has been initiated in the cell? What is this response, and how does the receptor binding to the signaling molecule cause its initiation? How does it start the response? First, understand that the receptor is just a protein. And like any other protein, it'll often undergo some kind of conformational change when its substrate molecule binds to its active site. Hydrophobic hormones, or those that are lipid-soluble, will pass through the membrane and bind with a free-floating receptor bouncing around the cytoplasm of the cell. In the case of gonadal hormones like estradiol and testosterone, the hormone receptor complex is moved to the nucleus, where it gets involved in up or down regulating the expression of particular genes. In cases like this, the hormone is directly responsible for initiating the response. Hydrophilic hormones, in contrast, won't move through the membrane. These hormones have to bind to a receptor on the cell's surface, which then reaches through the membrane or connects to proteins on the other side of the membrane on the inside of the cell. Because the lipid-insoluble hormones have to use these membrane proteins as intermediaries for their signal, they operate indirectly. Signal transduction occurs when the information in the signal molecule is passed through different mediums. In the first step, the information exists in a signaling molecule. The signaling molecule is the information. The fact that it exists is the information. After the signaling molecule binds to a receptor, it causes the receptor's conformation to change. The information now exists in the altered conformation of the receptor molecule, and this gets passed through to any protein that interacts with the altered receptor on the inside of the membrane. This might activate another protein, which activates another, and so on and so forth. And in this way, the information gets passed along this chain of proteins. The receptor mechanisms can be very complicated, involving dozens of different proteins that all interact together to carry along this information. A protein at any step in this process may activate multiple proteins, which in turn activates more than the single protein ever could. This effect, where the signal is given to multiple proteins and they all begin their own cascades, is called signal amplification, and it strengthens the response. There's two major types of signal processing systems called G proteins and enzyme-linked receptors. Each type of system shows tremendous variety across cells in an organism, and even more variety between species. But there's a couple similar features that exist between all of them. Some membrane receptors are connected to molecules called G proteins, which are studded in the membrane like anchors. The G proteins bind to GTP, a molecule that's similar to ATP, except it possesses the nucleotide guanine instead of adenosine. So when GTP binds to a G protein, it causes a major conformational shift. This shift causes the G protein to disassociate from the receptor molecule, and then to go ahead and associate with a specific enzyme. This GTP-G protein enzyme complex will then catalyze the synthesis of a new molecule called a secondary messenger. These secondary messenger molecules are small, and they're able to diffuse through the cell relatively rapidly. In this way, the GTP-G protein enzyme complex can boost the signal from a single hormone molecule into thousands of secondary messengers, which interact with various molecules in the cell to initiate a response. This is a textbook example of signal amplification. Secondary messengers like cyclic adenosine monophosphate activate particular protein kinases, molecules that catalyze the phosphorylation of other molecules. Other secondary messengers like calcium ions bind to a receptor called calmodulin, and the calcium-calmodulin complex will then bind to and activate or deactivate various proteins. Now I know this might be confusing, but it's very important to understand that the same type of secondary messenger molecule may induce a different kind of response in a different type of cell. Furthermore, a cell's response may require the actions of multiple different secondary messengers. Let's go back to that Song of Ice and Fire analogy that I used earlier. The white raven sent from the citadel is like a signaling molecule. The raven landing in the rookery is like the signaling molecule binding to its receptor. The bird keeper is like the G protein, because it's attached to the rookery receptor. The bird keeper/G protein is stimulated with GTP when he receives the message from the white raven, and goes on to find a nobleman in the castle. So let's say that our bird keeper/G protein finds the Lord, the ruler of the castle, and our enzyme. The bird keeper gives the message to the Lord, and in doing so, he becomes much like the enzyme G protein GTP complex. The Lord of the castle will then give the news to all of his subordinates, his advisors, his servants, his officers and administrators, and much like the secondary messengers, these people will take the news to their departments and organizations and get to work preparing for winter. This is essentially a medieval version of cellular signaling. Now, the other type of signal processing system involves receptors that are directly linked to the enzymes. When the receptor binds to a hormone, the enzyme changes its conformation, and the altered receptor-enzyme complex is then able to be phosphorylated, or phosphorylate itself, which will then attract a G protein. In this way, the enzyme-linked receptor process works in the reverse direction of the G protein process. The G protein is activated by the receptor-enzyme complex, which causes the phosphorylation of a different protein, a protein kinase. This protein kinase then causes the phosphorylation of another protein, which does the same to another protein, and so on and so forth in a phosphorylation cascade. In many cases, one kinase can activate multiple kinases, which causes signal amplification. There's one more step involved in cell signaling, and that is shutting the signal off. It's all well and good to send a signal when you need to, like a developmental hormone released at a specific time in the fetus, or the surge in sex hormones that's experienced during puberty, but it's equally important to shut down this signal when necessary. Failure to turn off a signal during fetal development can lead to gross deformities and health complications, if not outright death. Interfering with one or more proteins involved in the processing and amplification stages can usually downregulate or shut off a signal. For example, phosphorylation cascades can be extinguished with molecules called phosphatases, which dephosphorylate and thus deactivate many different proteins that are involved in the cascade. Secondary messengers can be metabolized and broken down or cleaned up, like when the smooth endoplasmic reticulum cleans up and stores calcium ions. Breaking the GTP on a G protein down into GDP will deactivate it and prevent it from binding to an enzyme to create more secondary messengers until it's been reactivated. In all of these ways, a sufficient effort to dephosphorylate proteins in a phosphorylation cascade, or to clean out secondary messengers, or to deactivate G proteins, all of these things can lead to a cessation of the signal. Perhaps the most famous example of a signal failing to be stopped is cancer. Tumors are caused by cancerous cells that cannot turn off the signal to keep dividing. The cancerous cells will divide, and divide, and divide, and divide, consuming resources and ever growing into a larger mass of cancer cells. A benign tumor is one that's been encapsulated, and its growth has slowed down or stopped. A benign tumor doesn't spread. Malignant tumors, on the other hand, aren't encapsulated. They replicate aggressively, and they do spread. Chunks of malignant cancerous cells can break off and float through your bloodstream, or they can get lodged in other areas of your body and begin reproducing to form new tumors. A benign tumor often looks like a lump or a growth, which can usually be removed with surgery. Malignant cancers, on the other hand, can form tumors almost anywhere in the body, requiring chemotherapy to treat. As there can be dozens of malignant tumors scattered all over the body, surgery is far too brutal and invasive of a method to be used to remove them, and that's why we have to use chemotherapy. Being able to stop a signal or downregulate its amplification are really important aspects of response regulation. Cells respond to various signals almost constantly. They respond to many different kinds of signals and hormones at the same time, processing them and performing the necessary responses all simultaneously. In many cases, the G proteins, enzymes, and the secondary messengers in one signaling pathway can interact with those in a different signaling pathway by up or down-regulating their expression. A hormone can bind to a receptor and activate an enzyme in a particular signaling pathway. This enzyme could be used to continue the pathway, or it might be able to inhibit some protein in a different pathway. This would reduce the signal strength of that second pathway, even though the initiating molecule is still present. Alternatively, the secondary messenger in one pathway might stimulate some protein in a different pathway, and that will encourage a stronger signal response in that different pathway. As each signaling pathway involves numerous steps, this gives numerous opportunities for various pathways to regulate one another because they can interact at these various steps. Now, because the cells are always being sent some kind of signals, there's always some kind of interaction between the active signaling pathways. Each pathway will perform its particular functions, including using its own products to regulate the performance of its own and other pathways. This crosstalk between signaling pathways is responsible for sustaining the health of the cell in a dynamic and very carefully balanced way. Even small fluctuations in the concentration of a signaling molecule can have enormous downstream effects for the cell, or the tissue, or the organism as a whole. Well, that should cover most of the very basics of cell-to-cell interactions. We started with cell walls and extracellular matrix, then junctions and desmosomes, and ended with hormones and signaling pathways. Before I end the episode, I want to very briefly discuss a really fascinating thing called quorum sensing. Now, all of the interactions that I described in this episode exist between multicellular organisms. They exist between the cells in one large multicellular organism. But then you have colonies of single-celled organisms that can interact with each other through quorum sensing. In these colonies of single-celled organisms, single-celled microbes, small molecules are released into the environment, and they diffuse to reach proximal individuals. In this way, colonies of single-celled organisms, particularly densely packed colonies, can communicate their situation and coordinate their activity in response. Quorum sensing allows colonies of single-celled organisms to behave in a manner that's eerily similar to the tissues of a multicellular organism, because they're all in constant communication with each other because of this diffusion of particular signaling molecules. Quorum sensing even allows cells of various prokaryote or protist species to engage in complex group behaviors, like moving across a surface or even arranging into a fruiting body. How cool is that? Alright, now I think that's pretty much everything. This was a fun episode, and I hope you enjoyed listening to it as much as I enjoyed doing the research and writing the notes and making the recording. I certainly hope that you learned something cool, something that you can talk about at the dinner table, or with your roommate, or to your dog if you have no one else. And at the very least, if you're taking a biology class, I hope that some of this information might help you pass your tests and understand the material a little bit better. And as always, thanks for listening.